Everybody ready for a big study weekend? This is your chance. Yeah, there we go. I like this. This is your chance to move up your grade, right? Let me turn that volume down just a little bit. Okay, so it's Dad's weekend. How many dads do we have here today? Oh, wow. Welcome. Have they been telling you how wonderful this class is?
Oh, yeah.
No, they haven't either. Well, I'm very happy that you're here. And in honor of your being here, we have a little something we like to do to welcome dads. Now, the rule is that the students have to really, this is a little thing I wrote for dads, and I want the students to really belt it out for dad. So even if your dad's not here, I want you thinking about dad as you're singing this song, okay? This is a song to the tune of an old Irish melody called Danny Boy. Okay? All right? So it's a very soulful, very mournful kind of song, and I want you to just give Dad everything you got here, okay? Everybody ready? Okay. Oh, Daddy dear, this is my biochemistry. The problem's long, the course is really tough. That last exam, it really put the fear in me. I studied lots, I hope it was enough. But let's forget about it while you're visiting and have some fun when we get out of class. Then when you're gone, I'll go back to my studying. That Kevin Ahern really, truly is an... I heard the wrong word there. I know I heard it there. So welcome, Dad. Happy to have you here. Get out of this. Okay. Now we have to do real things. You guys are having a good Dad's weekend so far? Up until that song? Okay. So last time I finished talking pretty much about signaling through the epidermal growth factor receptor. And I'll remind you of that process here. And the epidermal growth factor receptor plays a very important role in communicating that information from outside to inside that, hey, it's time to divide. Okay? A very important message. Now, you could imagine that if we screw up that system, we might have uncontrollable growth. And in fact, that does happen. Okay? It does happen. Okay? We saw, for example, how I described to you how a single point mutation in RAS could leave RAS in the permanently turned on state, which would now tell the cell to continue to divide. I need to tell you just a little bit more about RAS and give you a couple of terms. Okay? So as I said last time, RAS, when it's mutated, doesn't get turned off. It's left in that turned on state. It can't break down GTP. And as a consequence, it causes uncontrolled cell growth. I call the mutated form of RAS an oncogene. Okay? Notice I described the mutated form of RAS as the oncogene. All right? Well, what do we call the unmutated form of RAS? Why do cells have something that's going to cause them to divide uncontrollably? They don't. The normal form of RAS performs an important role in that signaling process. So we have to have a name for the normal form of RAS, the unmutated form of RAS. And the name that we give it is it's called a proto-oncogene, which essentially means a precursor of an oncogene. So long as RAS hasn't mutated, it performs its function very well. It tells the cell to divide when it gets the signal, and then it turns itself off, and that signal is gone. So, in general, and there are several hundred oncogenes that are out there, The unmutated forms are called proto-oncogenes, and the mutated forms that cause uncontrolled growth are called oncogenes. I want you to know the distinction between those two definitions. Okay? A proto-oncogene versus an oncogene. Well, let's imagine, if you will, that I mutated the epidermal growth factor receptor. And it started continually phosphorylating itself. Do you suppose the proto-oncogene receptor would be an oncogene? The EGF receptor would be an oncogene. I'll say that again. I kind of screwed that up, didn't I? Do you suppose that the mutated form of the EGF receptor under those circumstances would be an oncogene? Yes, it would. Okay? Because it's mutated, and a mutated form is, of course, the oncogene. In this case, the EGF receptor would be a proto-oncogene, the unmutated form that's doing its normal function. Okay. Now, there's quite a wide variety of proteins that play similar roles in cells in terms of controlling decisions about to divide or not to divide. And when they get mutated, they cause problems. I'm going to give you a couple of examples today, and also give you a couple of examples about how we have strategies for dealing with at least some of them. Okay? So we clear on what oncogenes and proto-oncogenes are? Okay. Well, there is another membrane receptor that is similar to the EGF receptor. Its function isn't fully understood, but it has a name. The name of this receptor is called HER2, H-E-R, as in his and her. All right? The HER receptor is capable of interacting with one of the monomers, remember this is a dimer, with one of the monomers of the EGF receptor and causing it to start signaling. And it appears that the normal function of the HER receptor may be to do just that, to help turn on this process at a low level in cells. Okay? So when the HER receptor binds to the EGF receptor, it doesn't require EGF, but it binds to it and starts this signaling process. So at a low level, this happens in normal cells without any consequence. In some, yes, ma'am.
Does the HER2 structurally look like EGF?
Does HER2 structurally look like the EGF receptor? And the answer is yes, it does. It doesn't look like EGF, it looks like the other receptor. It resembles the other half of the receptor. Okay? So the HER2 receptor resembles the half of the EGF receptor, which is how it's able to form a dimer with it. All right. Now, in the normal cell, that goes on at a low level, everybody's fine. If I have a cell that has had a mutation such that instead of making a normal amount of HER2, it starts making a lot of HER2, Houston, we've got a problem. And the problem is that this signaling starts going like crazy, and you can imagine what a lot of this signaling going on is going to do. It's going to cause the cell to divide when it shouldn't be dividing, and the signal doesn't get turned off. Because there's a lot of HER2, it's constantly signaling. We've also created an uncontrolled division, uncontrolled growth of a cell. This type of mutation happens in a variety of cancer cells. One cancer cell that it's commonly found in is breast cancer cells. Okay? It's commonly found in breast cancer cells. All right? Now, the good news is that there is a very interesting and fairly successful treatment for this that's been invented. And that invention involved creation of what's called a monoclonal antibody. So we talked about antibodies. Antibodies are simply immune system proteins that recognize specific structures. And a monoclonal antibody is a little bit fancier. We don't need to go into the immunology of it to understand it today. But suffice it to say that the monoclonal antibody will bind to the HER receptor. This monoclonal antibody has a name. It's called Herceptin, H-E-R-C-E-P-T-I-N. What Herceptin does is it effectively binds to that HER receptor and stops the signaling process. It's very effective. cancers that arise from overexpression of HER are very treatable by Herceptin. There's very little side effect. Okay? Many anti-cancer treatments have side effects. There's very little side effect associated with Herceptin, and it's very specific. That's probably why there's very little side effect that happens. Okay, so that's a very positive thing and a very cool way of treating certain types of cancer. Does anybody know anybody who's ever been treated with Herceptin? It's not uncommon anymore. No? Okay, maybe, okay, yeah. So, okay. That's what I want to say there. Any questions about that? Okay. Well, there's one more cancer I want to talk about. And it's actually this last one listed here on the sheet. This one is an interesting type of cancer that causes a certain type of leukemia. Okay? The acronym for it is CML, but that doesn't really matter. The most important thing is that we have a leukemia. And this leukemia arises in yet a different mechanism. Okay? ABL is a tyrosine receptor kinase, like we've seen before. Okay? It participates in a signaling process that stimulates cells to divide. Okay? So we can think of it in similar terms to, say, EGF receptor. Okay? EGF receptor was a tyrosine protein kinase. That's how it got all those phosphates on there. This guy is also it's ABL is right there. Okay? It's also a tyrosine kinase. Puts phosphates onto tyrosines. That's an important part of the signaling process that this protein's involved in, and as I said, it stimulates cells to divide. In the normal scheme of things, ABL is produced at a relatively low level in cells. And at that low level, it really keeps the cell in balance, when to divide, when not to divide, etc. There are a certain number of cases where it appears that ABL becomes joined to another gene called BCR. And this can happen as a result of a process called recombination. We'll talk about recombination next term, but it basically involves joining different DNAs together. These guys are on different chromosomes to start, but after this recombination has happened, the BCR becomes fused to the ABL. Now, the problem is that the control region for the BCR gets linked to it as well. And BCR is made at a much higher level than ABL is. So the fusion makes a heck of a lot of this fusion protein. All right, so we're together so far. We've created a fusion, and this fusion makes a lot more of the fusion than we had of just ABL alone. All right. So the problem is this fusion still has the tyrosine kinase on it. Well, just like we saw before with the other receptor, we make too much of a signaling receptor that's telling the cell to divide too much. This fusion is also going to tell the cell, divide, 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 divide. We've got a problem. Okay? This recombination is not an uncommon event. It does happen. If you get this fusion and nothing else happens, you're going to develop a deadly type of leukemia. That's the bad news. The good news is that there's a very good treatment for it. It's actually invented by somebody up at OHSU. Okay? Invented by somebody up at OHSU. And the treatment takes advantage of the fact that this is a tyrosine protein kinase. What if I put an inhibitor of tyrosine protein kinases into cells? What if I treated cells with tyrosine protein kinases? Would I stop that uncontrollable growth? That's not an unreasonable thing to ask, given what you've learned about tyrosine protein kinases. Well, that was the question they asked, and the answer they got was, it works pretty darn well. Okay? Again, low levels of nausea and other side effects associated with the treatment. And the beauty of this, and this, by the way, this compound is called Gleevec, G-L-E-E-V-E-C. The beauty of this compound is that it's fairly specific for this fusion. it doesn't muck with a lot of the other tyrosine kinases that are out there. It's probably, again, one of the reasons why it has limited side effects. This treatment is very effective for treating certain types of leukemia. Okay, so that's something that you can now take and learn. You've learned something about signaling, and as a result of learning something about signaling, you've also learned mechanisms or simple mechanisms of carcinogenesis and also simple treatments of that. So these are very, very powerful things for us to have. Okay, questions about that? Yes, ma'am.
Since this is a result of a chromosomal disorder, does that mean that the Gleevec has to be re-administered throughout life to avoid the leukemia?
That's a very good question. Because this is the result of a chromosomal disorder, does this mean that we need to have this for the rest of our life as a result of leukemia? Well, I don't have a complete answer to that question, but the simple answer is no. Because what you're aiming to do are kill the cancer cells, and the cancer cells are those that have this. Right? So by killing the cancer cells, you're actually eliminating those that have happened. That is, these aren't widespread throughout the genome, or everything would be a tumor at that point. Okay? Now, there may be follow-ups that people have to take. I can't comment on that. I don't know. But in essence, you're trying to knock out the cancer cells, and they are the ones that have suffered this mutation, or the recombination. Does it make sense?
Yeah.
Good. Other questions? No questions from dads, I guess. I always give dads pop quizzes, you know. Would you guys like that? Hey, Dad! He does this all the time. Okay. Well, that's what I want to say about signaling. So if there are no further questions, I'm going to turn our attention to a very important topic known as metabolic control. And I'm going to go through this fairly quickly because you've seen some of it, and the TAs are going to be working some problems for you in the class. There are also videos of me working some delta G problems for you that I will strongly encourage you to look at. Okay? So I'm not going to spend a lot of time in here going on calculations, but there are videos out there of me working problems. If you have questions, of course, as always, come see me. I'll be more than happy to try to answer your questions. Well, I said earlier in the term we would be giving some consideration to delta G because we have to be able to understand and predict directions of metabolic reactions. And directions of reactions are directly a function of energy. So we have to understand energy in order to understand whether a reaction goes forward under a given set of conditions or it goes backwards. If we think about the breakdown of glucose, which is something we're going to be talking about very shortly, we think about the breakdown of glucose. In the forward direction, we're breaking it down. In the backwards direction, we're making it. Okay? So being able to determine whether we're going forwards or backwards really tells us, are we breaking down glucose or making glucose? We need to be able to predict whether or not we're doing that. And the answers to those questions arise from an entity known as the Gibbs free energy. Now, you had Gibbs free energy in freshman chemistry, and you also had it very briefly at the beginning of this term. So I'm going to spend just a little bit more time talking about it and give you some examples about how Gibbs free energy is important. Okay? All right, so everybody has probably heard the equation delta G is equal to delta G 0 prime plus RT times the natural log of the products over the reactants. Okay? That's sort of schematically shown down here. And when I draw these with very simple reactions, A going to B. The more intermediates we have, the more complicated that reaction gets. And for our purposes, we're keeping this in simple terms, thinking about really what's happening. Okay? You all probably also memorized from freshman chemistry that if the delta G is negative, the reaction goes forward as it's written. If the delta G for a given reaction is positive, it goes backwards as written. And if the delta G for a reaction is equal to 0, then the reaction is at equilibrium. I pointed out to people earlier in the term that equilibrium does not mean equal amounts of products and reactants, contrary to what a lot of people like to think. It simply means that we have reached a point where the forward and backwards reactions are going the same so that the concentrations do not change. For a given reaction, that might be 5 times as much product as reactant, or 5 times as much reactant as product, or 2 times, or some number. Okay? We can actually calculate that number using some of the information that we will have. Okay. Well, delta G is related to a quantity known as delta G 0 prime. Okay? And in my experience in working with this with students have a lot of confusion about the difference between these two. And I hope to dispel that for you. All right? So delta G is the change in Gibbs free energy for a reaction. It refers to any set of conditions that I apply. Any. Okay? Now, when you studied the gas laws, for example, in freshman chemistry, you had something called STP, right? What was STP? Standard temperature and pressure. Those were standard conditions. Under STP, you could talk about 24.4 liters per mole of a gas, right? So not surprisingly, we're going to have STP for Gibbs free energy. Okay? If we measure it, if we measure the change in Gibbs free energy under STP, standard temperature and pressure, okay, then what we have is the delta G0 prime. So the delta G0 prime is the Gibbs free energy only, underline, only under standard conditions. Now there's more than temperature and pressure in standard conditions. One of the conditions is 1 molar products and reactants. Okay? 1 molar everything in a reaction. Now we'll see the significance of that in a minute. The reason we talk about delta G0 prime, you probably didn't get the prime part when you took freshman chemistry, is because for biological systems we have to make a little asterisk because we don't want protons at 1 molar. That would be a pH of 0, nothing would be alive. We couldn't study our reactions. So the asterisk is that little 0 prime that's there, and that prime refers to the fact that it's a biological system and we're studying it at pH 7. Okay? That's what the prime means. We're studying it at pH 7. So the standard temperature and pressure for a biological system, okay, we calculate it differently than we do for a different system. All right. Now, I've just told you that the delta G0 prime is the change in Gibbs free energy under standard conditions. 1 molar everything, blah, blah, blah. Okay? Let's plug that into our equation. We don't need to know what the temperature is. We don't even know that an R is the gas constant. It's just a constant. We don't even need to know what that is for this calculation. We simply need to know that we have standard conditions. Under standard conditions, B is at 1 molar and A is at 1 molar. What's the ratio of B over A? 1. What's the log of 1? 0. Whether it's a base 10 log or a natural log, it's always 0. So what happens to that term on the right? It disappears, right? Now you see why delta G equals delta G0 prime at standard conditions. It's not hard. Okay? All right. Now, what if I don't have standard conditions? What if I have 3 times as much B as I have A? Okay? If I have 3 times as much B as A, what's the value of the log term? Positive or negative? It's positive, right? Because I have a number that's greater than 1, that log term will be positive. Is the delta G under these conditions higher or lower than the delta G0 prime? It's higher because I'm adding a positive number to it, right? Makes sense? Let's imagine that I said, okay, I have the system at equilibrium and there's 3 times as much B as A. I want you to tell me if the delta G0 prime is negative or positive. The system is at equilibrium. What does that mean about delta G? It's 0. 0 equals delta G0 prime plus what? Or minus what? Is it plus or minus? It's plus, right? So the value of delta G0 prime, therefore, is negative. I have to add an equal negative number to a positive number to get 0, right? Now, we didn't punch anything in a calculator. You guys probably get tired of me hearing me say that. Didn't punch anything into a calculator. We were able to determine something about this value just by knowing values of logarithms. Okay? Makes sense? Clear as mud? You guys are back to your quiet mode. Well, delta G is a very, very important quantity for us to understand. I have an exercise, I have an example I want to give you here, okay? And this example is something that you can relate to, I think, okay? And the example is this reaction right here, okay? Let's see if I can scroll it up on the screen so it gets a little higher, okay? It's the best I've got. Look at this reaction. This is a reaction that happens inside of our body. Creatine plus ATP gives creatine phosphate plus ADP. Okay? Let me tell you a little bit about this reaction. Creatine phosphate is a molecule that's in our muscle cells, and it's in our muscle cells kind of like myoglobin is in our muscle cells. Myoglobin in our muscle cells, what was the function of myoglobin? Storing oxygen, right? So when the oxygen levels got low, then myoglobin started giving up oxygen, so the muscle cells had oxygen. Creatine phosphate does a similar thing. Creatine phosphate is a very high energy compound. Very high energy compound. When cells start running out of ATP, they use creatine phosphate. Now, I hear a lot of people take creatine and things like that, and I think it's one of the stupider things that you can do. I'll give you one example in a minute, okay? But suffice it to say that creatine phosphate is important for our muscles. When I take off and I go off on a 100-yard dash, A, I'm very slow, but B, very quickly my ATP levels start falling. If I don't have a battery backup real quick, I'm going to collapse before I get to 100 yards. You would too, even if you're not an old guy like me, okay? Because your ATP stocks go down very quickly. Giving something to back them up is very important, and that something that backs them up is creatine phosphate. Well, there's a problem with this. Creatine phosphate is a very high energy compound. In fact, the reason that your muscles use it is because it has more energy in it than ATP does. There's more energy in creatine phosphate than there is in ATP. Yet look at this reaction. The reaction says that I can make creatine phosphate starting with creatine and ATP. How can I do that? How do I make a reaction go forwards to make something that has even more energy than what I started, than the thing I started with? This is a pretty cool trick. All right, the answer is rooted in the Gibbs free energy equation. Right? What influences delta G? The ratio of B to A. Right? B being the products, A being the reactants. If I have a lot of A, what happens to that ratio? It gets smaller, and the natural log of that ratio becomes more negative. So I can drive this reaction forwards by putting a lot of creatine and ATP there. Right? I'm making the A term much larger. I'm therefore making that logarithm more negative, and eventually the reaction will proceed forwards. This delta G that we see, delta G0 prime that we see right here, tells us just that. Notice that delta G0 prime says it has a value of plus 12 kilojoules per mole. It says that at equilibrium, and this is something I'll probably ask you to calculate on an exam. It says that at equilibrium, I will have more reactants than I will products. That makes sense? How would that happen? Equilibrium, delta G is equal to 0, right? 0 equals 12, plus 12, plus the log, in this case, of B over A. RT times the log of B over A, right? What does the value of of the log term have to be if delta G0 prime is 12? It has to be negative 12. How do we get that term to be negative 12? By making A, there be more A, right? So now the equation is consistent with what we just sort of logically worked our way through. We have to have more A in order to make creatine phosphate. Now, that's a delta G exercise. Let's think about it in terms of a real-world exercise with respect to our muscles. Okay? I'm sitting around, eating pizza, drinking beer, watching the tube. Something I don't do, but I drink beer at least, okay? Sitting around drinking beer, eating pizza, watching the tube, all right? What's my level of burning up of ATP? Am I using a lot of ATP? Depending on what I'm watching on the tube, probably not, right? Okay. Whoa. You guys have dirty minds. God. Okay. My ATP levels are low, okay? Low, I'm sorry, they're high, whatever it is. Okay, I'm not burning ATP, so my ATP levels are high, right? I'm sitting here, I'm going to get in trouble. All right. My ATP levels are high. When my ATP levels are high, what happens to this reaction? Okay, it's going to go to the right? And I'm going to make creatine phosphate, which is good. So it's going to be there for that rainy day when I decide to get up and go for my 100-yard dash to the refrigerator to get more beer, right? Okay, and my ADP levels, I'm going to be making ADP from ATP, right? Okay, so I'm sitting there doing nothing. This is going to happen. Now I get up and I decide the house is on fire, and I better get the heck out of there. I've got a long house. I've got to run really fast and long to get out of there. And the first thing that happens when I get up and I start running is what's going to happen to my ATP levels? Doing. As ATP levels go down, what starts to happen here? The reaction starts moving to the left. And creatine phosphate kicks in and starts making ATP. Okay? So my system is set up such that that battery is letting go its charge when I need it. It's very cool. Okay? Now, people take creatine because, oh, it's going to increase athletic performance. As if increasing athletic performance made you more healthy, which it doesn't. Don't kid yourselves. Okay? Now, I'm going to take some creatine. I'm going to take some creatine, and I'm going to go out and run a race. Because I'm going to really be in good shape to do this. Right? You've heard this. Let's run that equation through. All right, so I take a lot of creatine just before my race. What's going to happen if I go and gobble a bunch of creatine before I decide to go out and do a 100-yard dash? What's my ATP levels going to be when I start? Low. Is that a good idea? No. Okay? Because I'm going to be totally dependent upon this thing going backwards to get me going. This is my backup. This should not be my main supply of energy. Yeah, I like that, huh? Okay? Yes. All right. So that's why some of these things are just really not a good idea. All right? Now, but what if I took creatine phosphate? Okay? What if I took creatine phosphate? That's the key. I got to take creatine phosphate here. If I take creatine phosphate, I'm going to increase my cellular levels of creatine, and I've already said increasing your cellular levels of creatine over long term probably isn't a good idea. Okay, you get the idea? Okay, so questions about that? No questions? Yes?
So, you didn't really go over why taking creatine phosphate was a bad idea. You were saying that
It's going to increase my overall concentrations of creatine,
right.
So that the next time I go out to run, what's going to happen?
I see.
Yeah. Okay. Now, there are people who claim this works, etc. I'm not going to argue with them, but I will argue with them from the point of view of this equation that it's probably not a good idea, and in general, taking things for athletic performance like this probably aren't good ideas either. Okay? Okay. Since nobody argued with me there, I've decided to give you another song today. So, and this is also for Dad. So, what's that? Oh, question, yeah. So the backwards reaction is simply there to make ATP. That's the only thing it's there for. Good question. Okay, so as I said, Dad's here, so we're going to have we've never done this before. We're going to have a second song, and I think you're going to know the melody. Okay? It's all about delta G, and please join me in singing to the same tune we just sang. Oh, delta G, the change in Gibbs free energy can tell us if a process will advance.
'Cause if the value's less than naught, it translates that reverse reactions haven't got a chance. But when the sign is plus, it is the opposite. And then the backwards happens all the time. A factor is the standard Gibbs free energy. So don't forget about the delta G naught prime.
Okay, that was good. Yep. All right. Now, we're actually moving along fairly well here. Maybe if you're really good, I'll let you out early today, but we'll have to see about that. So, okay. Now, we're getting ready to move to a section of the course involving metabolism. I've been talking about it, and we're getting closer to doing that. Metabolism is a very central part of biochemistry. We can think of metabolism as the chemistry of biochemistry. The sum of the reactions of metabolism are what give us life. Okay? The sum of the reactions of metabolism are what give us life. All right? Metabolic energy comes from, essentially, from oxidation. Unless you're a plant, in which case photosynthesis kicks in. For the most part, we certainly don't have photosynthesis. For the most part, life on Earth happens as a result of oxidative metabolism, with the exception of photosynthesis. Photosynthesis, of course, is the ultimate source of all metabolic energy on Earth, so it's not a trivial thing. Okay. We see continually that everything involved in metabolism is a swap between ATP and ADP. ATP being the high energy form, ADP being the low energy form. We use oxidation in going from right to left. Oxidation helps us to produce ATP. Okay? We use that ATP for a reason. That ATP might be used for motion, like we're talking about running and jumping and all those sorts of things. We use ATP for moving things across membranes. We'll talk about this next term. It's called active transport. We use it for making things. ATP energy is necessary if I want to make, for example, a fatty acid, or I want to make an amino acid, or I want to make a nucleotide. I have to have ATP energy to do it. So just like building a house requires an input of energy, so too does the building of molecules require an input of energy. The last thing is signal amplification. You've seen a little bit of this already. This was the phosphorylation business that we've talked about. So ATP is important, and the only way I get ATP is by going along this bottom pathway. Okay. Oxidation, as I said, is the key to metabolic energy. It's because of oxidation that we get energy. Look at the delta G 0' values. The top one's in kilojoules, the bottom one's in kilocalories, so they're just a conversion factor between them. The difference between methane and formic acid. Okay? What this tells us is that the more reduced a molecule is, by the way, the molecules on the left are the most reduced, and the molecules on the right are the most oxidized. The more reduced a molecule is, the more energy it contains that can be released to do things that we want. Oxidation of methane is what we do in our furnaces. We burn methane, convert it and make carbon dioxide, so it keeps us warm. The energy we get off from that is directly a result of the energy that's contained within methane. The more oxidized something is, the less energy it has. Methanol is more oxidized than methane is, and you can see it has less energy. The lower negative means less energy. Okay? So, understanding that oxidation process is important for us. Here are two molecules commonly used for oxidative purposes in cells. Glucose is the first one we'll talk about. The second one is a fatty acid. Okay? Of the two, you ought to be able to look at this and tell me if they had equal numbers of carbons, which one would have the most energy. The fatty acid would, because it's more reduced. You see there's a total of 2 oxygens, right? Everything else is carbon and hydrogen. This guy already has 6 oxygens and 6 carbons. If I had 16 carbons, I'd have 16 oxygens. Glucose is not going to have as much energy as a fatty acid is, and the reason we store fat is for this very reason. Fatty acids are esterified to glycerol to make fat because this has a lot more energy in it. Okay. We're going to oxidize glucose. We're going to oxidize fatty acids as ways of getting energy. Now, it would be nice if we could preferentially oxidize fatty acids because I wouldn't have to constantly be doing this, right? Going on the diet. Unfortunately, the easiest thing to oxidize turns out not to be fatty acids, but glucose. Our body is set up remarkably well to oxidize glucose remarkably rapidly. Okay? I gave you the example earlier about adrenaline. What adrenaline was doing was dumping a lot of glucose in the bloodstream, and that was going to target tissues that were picking it up and now using that for energy, my muscles, for example. Well, why doesn't our body dump a lot of fatty acids into our bloodstream? A very important reason why. Fatty acids aren't very soluble in water. Glucose is very soluble in water. Water is the medium of our bloodstream. I can't, yes, I can move fatty acids through my bloodstream, but it takes a while. I have to actually literally package them up and put them into little bundles so they can travel in my bloodstream. That takes a while. Well, if a grizzly bear is chasing me, right, I don't want to be waiting for all those little bundles to get made up so I finally have the energy, because by the time those bundles got made up from fatty acids, the grizzly bear would have eaten me. Okay? So, glucose is a very important compound for us with respect to oxidation. Fatty acids are as well, but something else has to happen with respect to them. We're going to talk about a metabolic pathway very soon called glycolysis. And glycolysis literally means the breakdown of glucose. It's probably the most common metabolic pathway on the face of the earth. And here is the only oxidation that occurs in the entire pathway. This is the only oxidation that happens in the pathway called glycolysis. Okay? You see, an aldehyde is oxidized to an acid. That one thing right there is what happens during glycolysis. Further oxidation of glucose happens in other pathways, not in this particular pathway. Okay? This is the actual reaction that's involved. Okay? This reaction also illustrates an important point. It's with this point that I'll finish today. Give you guys a chance to go out and have some fun. All right? And that is that biological oxidations do not occur the way that oxidations out in the rest of the world occur. Okay? It sort of makes sense. Think about this. If I light a match, that's an oxidation. The match is burning. What's the temperature inside that match? Got a problem, right? I can't just have uncontrolled oxidation going on because it would kill my cells. My cells go through relatively small oxidations under circumstances that they can control the reaction. A, enzymes are important. But B, the oxidation is happening in a different way. Oxidation, you may recall from your organic chemistry, or hopefully from me earlier this term, occurs when electrons are lost from a molecule. Electrons are lost in an oxidation. Well, they don't disappear. Right? We're not doing nuclear chemistry, so we can't destroy matter. Those electrons have to go somewhere. And if we just let them out into the open, we have dreadful problems. We'll talk about reactive oxygen species next term. We don't want those electrons to just go flying freely. We want those electrons to be contained and carried by something, and they're carried by electron carriers. NAD is an electron carrier. It accepts the electrons that glyceraldehyde 3-phosphate is giving up. Glyceraldehyde 3-phosphate is getting oxidized, it's giving up its electrons, and it's donating its electrons to NAD. When it donates its electrons to NAD, NAD picks up a proton as well and becomes NADH. Whenever you see a reaction, NAD going to NADH, you automatically know an oxidation has occurred. Always. Always, 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 always. No exceptions to that. Because to go from NAD plus to NADH requires electrons. If I go backwards, and yes, I can go backwards, NADH is giving up its electrons. It's reducing this compound to make that compound. Okay? When I see NADH going to NAD, a reduction is always going on. All right. For every oxidation, this is the last thing I'm going to say, and I hear the shuffling of papers, and it's the most important point. Okay? For every oxidation, there's an equal and opposite reduction. GAP is losing electrons. It's getting oxidized. NAD is gaining electrons. It's becoming reduced. All right? For one thing to get oxidized, something else will get reduced. All right? I'll come back to that later in the term, but remember that that's an important point. Okay, have fun. Thanks for coming, dads. Oh, you are?
Yes, Lauren.
You're Lauren. Okay, I need to talk to you. So I'll need to talk to you in my office, but I can't right now. Are you free later this afternoon?
I'm actually going home this weekend for Dad's weekend. I'm leaving right after my 3 o'clock class.
Okay. Well, I need to talk to you. So you have to decide when we're going to do that. But I have a matter I need to talk to you about.